All right, hello, welcome. Another episode of Wild Wisdom and Storytelling with Herbal Elder Blaine Andrusek.
Um, yeah, you like running with that, don't you?
Yeah.
As opposed to old retired witch doctor. Anyway,
Yeah, take your pick, right?
I'm really appreciating that as we've done some of these YouTubes, and I'm just getting out there a bit. How many people are getting a hold of me, and that I'm still playing the role.
Right.
I'm not getting paid for it anymore. But years ago, when I lived in the woods alone, the owls told me that my job was to never stop learning and to teach. That I
Right.
had a gift for that, and it feels real good just to continue doing
Yeah.
things like what we're doing together.
Yeah, awesome.
So anyway, you hear us talking at times about looking at a plant and kind of going, well, I think we have to key that out.
Yeah.
And I felt that a lot of people don't know what we're talking about with a key.
That's right. This is not car keys, this is not keying a car. Like this is
So
keying out plants.
There's an old
Keying out mushrooms.
Latin word, taxus, a little different than paying taxes. It's just taxus, and it means to arrange.
Right.
So the art of taxonomy is to arrange things.
Yeah.
Now, when I teach botany, on opening night, I always like to say, we're gonna learn a whole bunch of, unfortunately, Latin and a whole bunch of new descriptive adjectives. So studying botany is almost a language class more than a plant class.
Right.
And people say, well, why do we have to learn Latin?
Yeah.
And I go, well, because it's an old language that's never changed, and English is full of double meanings for things.
It really is.
But Latin, you can take a word like down, D-O-W-N, four-letter word, and it can mean so many things. Like, hey, I'm down with that. Or, wow, the sky's falling, get down. Or I'm feeling kind of down today. Or what do you think about this new down vest? Like, it's got so many meanings, right?
Yeah.
But in Latin, a word like arvensis means of pasture, or of the field.
Yeah.
Aquaticus is easy. It likes wet feet.
Yeah.
But there's all of these other words that are there. So Latin names of a species, the first word, and if you have a computer and can do it, Latin names are always done in italics.
Right.
The first word is capitalized, the second word isn't.
Yeah.
So the second word is a descriptive adjective, which could be about the shape of the leaves or the type of flower.
Yeah.
Or I found a new one that was never around before, so we might call it Violus Andruseki.
Right.
So when you see those two I's, that's how you say EI. Okay. Anyway, so we have these books where, like the one that I spent a lot of time with, and it was off market for a while, and Agriculture Canada brought it back, is AC Bud. And so that's a plant word already, right?
Yeah, very appropriate name.
It's AC Bud. So anyway, you can see how thick that is.
Yeah.
But if it's growing in Alberta, it's in this book.
Yeah.
But how do we fish through all of these pages, right?
Yeah.
So we have to somehow break things down. So there was a little game we used to play on the way to the cottage when I was a kid. It was like, I spy with my little eye.
Yeah.
So for me, it's sort of like you take a plant and you have to go, look at me, what do you see?
Right.
Right? And then that's where you have to learn a little bit of the taxon or the morphology of plants. There's some new words for all of the different shapes of leaves and what the edge of the stem looks like and how the leaves are attached. So there's a bit of that learning curve, but the keys always have a few pages. You just had one open there for some of the mushroom shapes, where there's just, I don't know if I'm going to get anywhere with this, but I'm going to stop there. So what you're doing is trying to break it down with a system, and there was, what was he, Swiss, I think, Lewis.
Okay.
Was it, I think that, did I write that down just because I haven't taught some of this stuff for a while? Going back to the 1700s. gosh, it doesn't matter. But he's the one who started this binomial system where we start with questions that break it down systematically. And I always like to tell my botany students this little phrase, mostly so.
Right.
People, especially men, want to put things in boxes
Yeah.
and straight lines.
Right.
And nature doesn't think that way, right?
No.
So all the rules, I said, we're going to learn a whole bunch of rules, and then sometimes we break them. Like rules are meant to be broken, right? So we can take, just at the beginning of a book like this, so this is a key. If it's a wild plant in Alberta, it's in here. How do we find it? So at the start, the first questions are, so we're going to start with something called divisions, and then we go to classes, and then orders, and then eventually families, and then genus, and then species.
Right.
And a species, by definition, is something that can reproduce and always look the same.
Okay.
So all the different breeds of dogs are the same species. They're just different varieties
Right. Okay.
of one species. Okay.
Yeah.
So a Chihuahua and a Saint Bernard can actually reproduce. They might need a ladder, but it would work, right? Okay.
Yeah.
They can pollinate, sort of thing.
Yeah.
So here's the first question: is plants without true flowers reproducing by spores?
Uh-huh.
Oh, or plants with flowers reproducing by seeds. Now, right there, it's going to lead us to reproducing by spores. That's going to lead us to another whole key
Right.
about mushrooms.
Yeah.
Right? So you always get a choice, and once in a while you'll realize you made a mistake and you might have to back up.
Right.
Okay, that'll happen. So we're going to go with plants with flowers reproducing by seeds. So right away, it says go to page 56. So we just left a whole bunch of stuff behind us. And we go to page 56. And we're into a division called spermatophyta, sperm, seed-bearing plants, or a subdivision, gymnosperm. And because we're Canadians, we say gymnospermae. So these are naked seeded plants. The seeds aren't in an embryo or anything like that. And
Right.
gymnosperms are things like all the conifers.
Okay. Yeah.
Right. So we want to go with seed-bearing plants. So with this, I thought that was going to throw me to. It didn't. So this whole first section is going to be the conifers. And where's my other little... So we keep just getting... Where did I leave that list that I had?
I think you just popped it in.
Look at
where you
me.
were with conifers.
Where do you see? So the next order is where we go from seeds, and then we're into a subdivision called Magnophyllophyta, and there's something called monocot or dicot. So that's a subdivision.
Right.
Now, that's going to cut all of the green plants in Alberta into one of two major groups. So every seed has a little baby plant in it. It's like an embryo.
Right.
And it's the most important thing is that the seed leaf, which is called a cotyledon, there's either one or two.
Right.
So if it's a monocotyledone, it's got a list of things that it subscribes to, like the plant parts, the flower parts, the sexual parts, petals and such, are always in threes.
Right.
Or combinations of 3, 6, 9,
Right.
etc. And that's going to include families eventually, like orchids and lilies and various things, where dicotyledones are going to have 4 or 5
Right.
parts too, like a wild rose has 25 stamen, and it's got 5 petals.
Right.
And under petals are subtending leaves called sepals. And then under that, if they might be something called bracts. So there's all these words that we have to learn. But when you look at it, it's interesting that plants have, like, you think about when you eat an apple, or just look at an apple core, there's little rooms there for all of the seeds.
Right.
Right? So those are the embryos. So before the flower on an apple tree even ever opens, the tree knows how many seeds are going to be in the core.
Right.
Just as, and this surprises a lot of women, don't even know that how many chances you're going to ovulate
Right.
is determined at a very young age. And then, you get to puberty, and somebody upstairs, where we deal with hormones and things, said, well, now, girlie, it's time to play cards. And hormones control the drip.
Right.
And you've got however many decades of chances to produce a new human.
Yeah.
Right. And that's all,
Predetermined.
predetermined.
Yeah.
Okay. So we've got these two subdivisions, gymnosperm and then monocotyledon A. See, it's for Canadians. And so we can skip through this pretty quickly. As we get to dicotyledonase, then right away we get three questions. Apetalous, flowers with only one floral ring, because it's apetalous, or corypetalous, flowers with two floral rings, or sympedalous, flowers with the petals wholly or partly united into a tube or bell. So you think about harebells and things like that, where there's bell flowers, right? So it breaks down really quickly, and then that'll tell you to go to page 29, and then you go to page 29. And the beauty of this is that if a flower book, if I see something called Wildflowers of Alberta, I want it to be based on this system.
Right.
Because after you learn a bit of botany, you know that the aster family, which is like sunflowers and asters themselves, are going to be at the back end of the book.
Right.
So you can skip through that much book, just to get to where the asters are.
Yeah.
And the rose family is going to be about in the middle.
Right.
And, it saves us a whole lot of time.
Yeah, a little bit cumbersome at first, but once you learn the system, then you can just...
So then you can look up the family is going to be called Rosaceae.
Yeah.
And then in the rose family, we've got things that are obvious, like the wild rose.
Yeah.
But you can skip in the book, you can just skip to the rose family, and then you get another whole key.
Right.
So with each of these subtitles, we get another set of questions. And you just keep asking questions until you look at, I think there's maybe 23 species of wild sage in Alberta. So you can just look up any of them and you get another key.
Right.
And you play 20 questions again.
Right. Yeah. Does it have this? Does it have that?
Yeah.
Yeah.
The shape of the leaves, etc.
Right.
Until you get to the point where you're looking at the species.
Right.
So it saves us a whole lot of time. Well, there was a harebell. But anyway, so as opposed to, I've seen wildflower books where they're arranged by the color of the flowers.
Oh, gosh.
Oh, I hate those because there's quite a few wild plants that come in three colors.
Right.
So where did this author decide to put the pink ones?
Yeah.
Or
exactly.
whatever,
And good luck if you're colorblind.
Yeah. So, or there's a lot of books, like back to mushrooms, I've seen mushroom books where they're not organized at all.
Right.
And whether you know a little bit, a lot, nothing, everything, well, nobody knows everything, but you could know a lot about mushrooms and you have no choice but to just flip through pages until you might randomly see the one you think might look like that and blah, blah, blah.
Yeah.
So we need to have things arranged. It saves you a lot of time, and it doesn't take that much time to learn how to use a key. So if you're serious about wild plants, some sort of key is a good thing to own.
Yeah.
Or a lot of the plants, like the little one you have there about mushrooms, does have its own key, or it'll start, just go to the groups there.
Yeah, exactly.
At the beginning,
So
it'll just have different shapes that are very obvious.
yeah. And again, it's asking, the first question is, does the mushroom have gills? If it does, follow along. If not, turn to the back and it doesn't have gills, then you got to go through a whole other series of questions.
Yeah, same thing, where it breaks down pretty quick.
Yeah.
So that's what taxonomy is all about. It's arranging things to save us time.
Right. Yeah, amazing. And similar to your Big Buds Flora, and you talk about, so here, this is the book, Mushrooms Demystified, that goes with, you got your handy little pocket guide, which, most people, they're probably, maybe they'll use the key. Otherwise, it's a bit of kind of flicking through. And, if we get into, like the agaricus species, and we're trying to decipher between that and amanitas, or even just the type of agaricus, sometimes you can do it with that book. Otherwise, you're much better off because it's just so much more detailed. Before we were just checking, okay, how many species of plants in Canada? Well, it turns out there's 5 to 5,500, somewhere in that range. With mushrooms, it's anywhere from 6 to 7,500 known species, and they're guesstimating 100 to 150,000 unknown species. So if you want to be very precise, like 23 different kinds of Artemisia, right? However many types of Armillaria or Agaricus or Lactarius, you really have to kind of dive in to this book in a similar way to really key it out.
Now, mushrooms were here long before we were.
That's right. That's what they say.
And I just found it fascinating when I first learned that a mushroom spore is the only thing on the planet that we consider a life form that's designed for the vacuum of deep space.
Right.
So as we go further and further out in the Earth's atmosphere, you go from house dust is everything from your own dead skin, to just leftover black pepper from supper last night,
Yeah.
all sorts of things floating around the house, including mushroom spores.
Mm-hmm.
As we go higher and higher and higher, you end up with the outer atmosphere. All that's still there and rising are spores.
Yeah.
So when you have a mushroom, let's just say, just hold the cover up there.
Yeah.
If you have a mushroom that size in real life, when that opens, that happens to be a guild mushroom, I think. When that opens, hundreds of thousands of little spores, each one is invisible to the naked eye, are going to fall out. And with just a little puff, they can float away in the breeze and go up and up and up and end up on the other side of the planet.
Yeah.
You might have a spore that started in Malcolm's backyard end up in Japan.
Right.
Now, lucky for us, they're very fussy where they will grow.
Yeah.
Because otherwise, just in the time we're shooting this little video, we'd have to use a shovel to get me home at the end. Like there's that many.
Yeah.
I just inhaled some. They're around everywhere, right?
Yeah.
So each mushroom spore wants to have maybe a particular grass or leaf or some sort of substructure at the right temperature, the right moisture content. And then that spore from Malcolm's backyard found just the right place in Japan and opened up and started a whole new life cycle, right? So just like when we pick an apple off a green tree, the seeds are meant to go somewhere you aren't.
Right. Yeah.
Right? It's like go west, young man.
Yeah.
Or whatever, right? So we have all these systems that green plants use, like samaras. The maple and elm trees have those little helicopters that fly across, or dandelions have little kites, right? And they can go across the whole lake.
Or the burdock.
You can, the burrs stick to the plant. Your genes or a dog or any sort of.
Yeah.
So we had plants that are common in Alberta now that were never intended to come here, but they had burrs, so they were stuck to the livestock. And then as the pioneers came with their livestock wagon behind the wagon trails, and the wagons made ruts in the ground, and the seeds were falling out of the sheep. Yeah. And now we have the plants growing all over the country, right? Like plantain. Mm-hmm. You've heard us talk about that elsewhere. Very useful wild plant. And the natives used to call it white man's footprints. Yeah. Because they'd never seen this plant before the settlers came. Right. And then, they followed us and so everywhere. So anyway, back to the shrooms now. Those spores can fly around end up just about anywhere. They're fussy where they'll hatch. They need just the right everything. But so part of learning about green plants or mushrooms is when you find something, I think with the green plants, the start for most people is to say they got their Flowers of Alberta book, and you're out on a hike and you see a cluster over there of these pink flowers, and you might go over there and look in the book and find, oh, this is called wild bergamot.
Mm-hmm.
And you can, if you once you learn a little bit, you rub your fingers on the stem, and it's got a square stalk.
Right.
And if you have a plant with a square stem, it's automatically in the mint family. Whether you're here in this yard or in K country or in South America.
Yeah.
You know, it's just, so we have these little, that's another characteristic of each plant family
Yeah.
or order, depending on how big we have to play
Yeah.
100 questions, right? It's just bing,
Yeah,
bing, bing,
asking
bing.
those questions. And one thing we wanted to do today was to show you within the kind of the kingdom of fungi, some of those questions might be, what color are the spores? And so Blaine is going to lead us through, give us a little background on like why use a spore print. But most importantly, we're going to show you how to do a spore print.
Okay, good. Now, depending on the ripeness, like when did the mushroom open?
Right.
We have things like portobello mushrooms, for example, which you can buy at Safeway or any health food store,
Yeah.
whatever. They grow wild here, at least now. They're a meadow mushroom, and when you get them, when they've just opened for a few days, you look at them. The bottoms are pink,
Mm-hmm.
but the spores are actually chocolate brown.
Right.
And eventually, if you have them laying around, whether they're in the fridge now or even just later in the field, I've picked them in parks in the city and people's lawns even.
Yeah, yeah.
And they're one of the mushrooms. I find it so fascinating how plants learn to do things. Like I have found fully grown portobellas on borders out on the street where they've learned to grow the stem on a mushroom is called the stipe. So normally the stem's about this long, and then there's the mushroom, but they've learned to duck the lawnmowers.
Yeah, yeah.
So we have a full-size portobello that's only got this much stem.
Yeah.
And this is part of what we call plant wisdom.
Mm-hmm.
You know, they learn to adapt. Right?
Yeah, for sure.
So once we have a mushroom, that's its fruiting body, and something stimulated it to bear fruit. One of the things is that we'll just touch on mycelium here a little bit.
Sure.
So when you pick a mushroom, say the cover size there, out of the lawn
Yeah.
or off a tree, some rotting tree, a log or something, that's like picking an apple off the tree. That's just its fruit. The real organism is typically invisible to the naked eye, and it's living inside the dirt or inside the rotting log.
Yeah.
So we've got parasites, saprophytes, and
The mycorrhizal.
So some fungi will actually kill a plant, but some of them just live on a dead thing.
Yeah,
They're part of
the decomposers, right? We
and
learned
those.
that.
Yeah, so they
Yeah.
break down and end up sort of fertilizing the soil around them.
And then you got the mycorrhizal, which are kind of symbiotic with the other plants. So they're exchanging nutrients with the trees and plants, and they play a role in the ecosystem that way.
Yeah, fungi have apparently every tree in this country has a relationship with the fungi and their fungals, and everybody gets along.
Mm-hmm.
And the fungus is capable of dissolving some of the mineral elements in the soil that the plant can't.
Yeah.
And some of the mycorrhiza will actually penetrate the rootlets of the tree And what the tree gives the mushroom is moisture
And sugars.
and frost protection too.
Right.
Because they're more sturdy that way. So it's a win-win situation.
Mm-hmm.
And we have things like when we go to things like, well, I'm getting ahead of myself here now on the birch trees, where people learn to actually farm the fungi.
Oh, right. Yeah, like with chaga.
With chaga.
Yeah.
So I had read somewhere, tell me if I'm wrong, that the chaga, the mycelium, like if you find one on a birch tree, the mycelium is actually quite large. It could be a block long,
Right.
growing in the ground. And on the root level, on the rootlet level, it gets inside the tree and under the bark. And one of the things it gives the tree, it's almost like a Band-Aid. If a branch breaks in the wind or you chop the branch off,
Mm-hmm.
chaga will grow to form a scab.
Right.
It even looks like a scab.
Mm-hmm.
So where they farm them in Russia, they've made these special axes where they go through a birch forest as they're gathering the chaga. They're doing damage to the trees so that they initiate the future
Right.
growth, right?
Yeah,
So
yeah.
they can actually sort of farm the fungi that way and manage it that way.
Yeah.
So it's a win-win with fungi. They were here before us, and they'll be here if we don't get our act together. They'll be here long after us.
Yeah.
I laughed the other day when I saw somebody wearing
Decomposing us all.
a shirt that said "Save the Planet," and I kind of laughed, and I said, "The planet doesn't need saving.
No.
Our silly butts might.
Yeah.
We might be completely written off and extinct,
Yeah.
but the planet will be just fine. It'll
Yeah,
recover.
exactly.
It's done it many times, right?"
Yeah.
Anyway, let's not go down that path. So
somebody was making a joke the other day about global warming. It's like, well, it's just the Earth going through menopause. That's
okay. Anyway,
yeah, she's got her own cycles,
um,
and we are small and insignificant in relation to the Earth here.
so some of these characteristics that you learn the words for, some of the fungi are growing underground, under the surface of the ground, in usually sort of an egg-shaped form. And if you slice them open, you can actually see a little structure in there.
Yeah.
Otherwise, it's a puffball.
Right.
And that's another whole group. So you go back to the key and
Yeah.
find the puffball section
Right.
and look for other clues. Right?
Yeah.
But if there's little things going on there, and you can see that it's a mushroom, I'm just going to grab this again.
Yeah.
So what happened is, is that showing?
Yeah.
So this was attached to that,
Yeah.
and it broke when the lid opened up. This separated, and then it's called a veil, right? It's like a little skirt. And sometimes when they break open down here, there's a cup still in the soil there. It's called a vulva.
Yeah.
And you want to be careful with stemmed mushrooms to, at times, pull a little bit of soil around from the edge of the stipe and make sure there's no vulva. And then we can look at other clues like this. And sometimes when that breaks open, little flecks, little pieces of the veil break. And that's when you get that classic, what people say, what's the difference between a mushroom and a toadstool? Nothing, really. They're just
Right.
words.
Yeah.
So it doesn't mean that one or the other is always poisonous. But when you get the little broken flakes, like oatmeal, on top of the red top, for example, that tells you it might be an amanita. And we've got some amanitas that grow in Alberta
Yeah.
that
we
are
do.
deathly.
Yeah.
One's called the destroying angel.
Yeah.
So you got to pay attention to all these little things like a veil. Now, kind of segue. So we've got a mushroom about this size, and one of the things that might have triggered the growth was that the mycorrhiza, I heard, it surprised me that it was on just regular common news one day that a group of mycologists working in southern Alberta found a mycorrhiza that was 10 kilometers wide.
Right.
Like it literally
Yeah, they
owned
can
a
be
small
huge.
town.
Yeah.
So all of the soil has it. So because they love moisture, if it's a day like today and it's gloomy and it's moist, that for the fungi and his fungal is like boom time. It's like, yeehaw, they love moisture and they grow and grow and grow.
Yeah.
And then all of a sudden it stops and gets hot really quickly.
Right.
They go, honey, this could be the end.
Right.
We might not make it, dear. We better, yes, dear, I agree. We better, and up pop mushrooms to save the species.
Right.
Just like Superman got blasted off his home planet.
Yeah, yeah.
Right? So a fruiting body is designed to get spores into the air to make sure the species doesn't die.
Yeah.
Right? And so back to a mushroom fruits in Malcolm's backyard, it ends up with a child in Japan that can spread spores again, some of which can go up into space and go all around the planet 3 times.
Yeah.
And it's just, that amazes me that they can live.
Oh,
They're
yeah.
like the original satellites, right?
Yeah. Well, and there's even theories that like spores were one of the first things that came to Earth to colonize it and have subsequently created the conditions where all other life has been able to exist and thrive within.
Yeah, and I agree with that.
Yeah.
Yeah, there's enough
Yeah.
evidence to support that theory. It's
Yeah. Okay, so when we're talking about identifying mushrooms and keying things out, does it have gills? Does it not have gills? As you keep going through, as you mentioned, right? Are the gills, are they white, pinkish, yellow? And if the mushroom is encased by a veil, does it have a vulva at the base? Like yes, no, you keep not going through. And then all at the bottom, if the spores are white, or in one case green, that means one thing. If the spores are rusty orange to brown or black, that means another thing. And so when it comes to
Right.
mushrooms,
Sorry.
doing spore prints can
yes,
be,
I got way off topic there.
yeah, can be really a key part of your identification process between this is a delicious dinner and this is deadly poisonous.
So one of the first things you're going to do when you pull up a mushroom like that,
Yeah.
for starters, if there's a bunch of little mushrooms somewhere on a log, I've never taken the time to bother dealing with them because they'd never give me supper.
Right.
And I could waste a whole day.
Yeah.
And when I started taking photographs for classes, then I would stop and photograph them and blah, 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 blah.
Right.
But unless there's a lot of the fruiting bodies,
Yeah.
enough to make it worthwhile, I wouldn't bother keying them out.
Right.
Like we've had times in the dog park where all of a sudden, overnight from here to the window, there's about 500 of a
Right.
mushroom, and everybody's asking me if we can eat them.
Yeah,
So
yeah.
it's not a mushroom I've seen before.
Right.
There's a few mushrooms that, in my years, I don't know, pick a number, but there's only about 200 mushrooms that I don't need a book to know.
Right.
Some of them are like the shaggy mane,
Yeah.
is very exclusive.
Yeah.
We get things like that. But so if you take the mushroom and turn it over, you're going to see one of two things. The bottom is either just, it looks like a sponge with little holes in it, or it's got gills.
Yeah.
Now, the part that you can't do without a microscope is look at, when you see the gills, they're obvious with the naked eye, but coming out of the sides of the gills are little things that look like clusters of grapes. And when the grapes open, that's when the spores drop.
Right.
And in a lot of cases, you would actually need a microscope, or some of them change color with dyes. That's all over my head. But the actual shape of the spore, there's all of these details that can go through. But a good part of it for normal people just to find edible mushrooms is just the color of the spore.
Yeah.
So if we see a gilled mushroom, then for the book, that one book that I was holding up, O.K. Miller died years ago, but his son apparently has picked it up. And I noticed online when I was checking a few months ago that this book is still available on Amazon. And he was just about finishing a second edition when he died, and I think his wife finished it up.
Right.
So there's a second one, and then I think his son has taken that over.
Yeah.
So I just like the way his key works for the gilled mushrooms.
Right.
That's why I loved that book. And I never would have got I didn't know anything about mushrooms when I was canoeing a lot when I was young. And I just, one day, up on the Churchill River system in Saskatchewan, you might get stuck on an island and it rains and big winds and you can't go fishing or anything.
Yeah.
You go out hiking and there's just hundreds of fungi. And they're so
Right.
fascinating, right?
Yeah.
Like there was one, I got up one morning, just crawled out of my tent to have a pee. And there was this thing that came up overnight. It was about 4 inches wide. And it just looked like a little piece of sponge. And it had this, it looked like cherry jelly. It's a particular mushroom that does that. It's one of the Boletus
Right.
that's known for, and so that's a very distinct characteristic, right?
Yeah.
So anyway, if you've got gills, you go to the section in the book with gills,
Yeah.
and then you take a spore print, and that gives you another whole key. So what mushrooms, what Malcolm's done, and we might have had some different colors if it was still mushroom season, but it's not. So because they're so tiny and fly away at the slightest bit of breeze, you have to take, and because they can be white, or like he was saying, they can be pink, different shades of rusty colored, or chocolate brown, purple brown. Purple brown are a good sign. Those are still acai. So purple brown's a nice color. But what happens is thousands of spores fall out, and it's almost like the negative of a photograph. And you get these little, they're like snowdrifts.
Yeah.
Now, white ones would be hard to see. You can see them, but it would be hard to see them on white paper. And you don't want red paper because white ones might look pink. So we want, and I was trying to find, but I didn't have them, to save them for classes. I've used lacquer just to spray these so that they don't smear anymore.
Right.
Like right now, if I could smudge those all over.
Yeah.
So the idea is if there's any breeze at all in the room, you're not going to get a spore print. So depending on how big the mushroom is, like some of those could have worked with a coffee cup.
Yeah.
You just need something big enough to cover them so that the breezes in the room don't, like if you had a ceiling fan or whatever,
Yeah.
or just opening the door, there's a little above
Right.
there. So you just need to cover the mushroom.
Yeah.
And sometimes you'll get a print in an hour.
Okay.
But overnight for sure.
Yeah.
And at times, I had, when I used to be canoeing and fishing a lot and whatever, I had made some little, I had little containers I used to buy bulk film in. It doesn't make Tupperware would do. And cut black and white circles, half and half, so that I had half the paper in each container was black, half was white.
Right.
And then with some crumpled up wax paper, so that if I found some mushrooms that were worth checking out, sometimes even by the end of a hike. The wax paper was just so they didn't wiggle around.
Oh yeah.
So
yeah.
sometimes even just by the time you get home. Or other times, here's a term, cespitose, that's spelt with a C. Some fungi grow in what are called cespitose clusters,
Right.
where from one mycelium in some bark, you might have this whole arrangement with about 30 fruiting bodies, and there's rust-colored spores that are dumping all over each other.
Right.
So you can see it has a rusty spore print without doing any of this. But if I don't know the species for sure, like a portabella, then I'm going to say I got to do a spore print.
Yeah.
So back to the dog park, there's all of these good-looking mushrooms. And when I come back the next day after doing the spore print and keying them out, I got to go, sorry, folks, we're not touching these. It's a category that may be okay,
Right.
but when I get into that maybe business with authors, there's only a couple mushrooms in Alberta that would actually kill you, but there's a whole lot that would make you wish you were dead
Right.
for a day or two.
Yeah,
You know,
yeah.
there's like diarrhea, vomiting,
Yeah.
intense cramping. You know, it's just not worth it, right? So unless
Yeah.
you're for sure. So the spore print then, this is a white print, then we go back to the book
Yeah.
and go to the section on white spore prints. Gilled mushrooms with white spore prints,
Right.
right? So all of the gilled mushrooms cumulatively are called Basidiomycetes. And when we get into the sponge-bottomed ones, they're called Ascomycetes,
Right.
but they're still dropping similar thing. Upside each tube is a little structure with sacs of spores, and they fall out. So you
Right.
can still get the color if you needed to.
Yeah.
But for gill mushrooms, it's quick and easy to do this. And there's a general rule. So when we go back to the divisions, it's different. It's, as soon as you have a sponge bottom, it's a type of Boletus, but the word Boletus can be an order and a family and a species.
Right.
So that you can have a Boletus, Boletus, Boletus.
Okay.
And as a general rule in the field, if you don't have any books with you, I've been told to, if it bruises or looks like it's, when you rub it, if it looks like it's dripping even red, looks like blood, if you don't have field guides, I wouldn't eat them.
Right.
But there's exceptions to
There
every
always
rule,
is.
like I said
Yeah.
at the beginning, right? So there's one genus called Lexinum,
Yeah.
which you're a big fan of,
Yeah.
and it actually does bruise.
Right.
When you handle it, it turns kind of a gray-purple color.
Yeah.
But on the stipe, it has all these little black
They call them scabers.
scabers, right?
Yeah.
Scabers, these little black things, and that tells you this is the you call them aspen conchs.
Aspen bolete.
Well, bolete, yeah.
Yeah.
So it's a Boletes lexinum, and it's okay that it bruises because it's got the scabers.
Yeah.
So there's all these little things you learn over the years.
Yeah.
But I've never done a spore print from a Boletus.
Right. Well, like you say, you don't always need to do a spore print. I think it's spore prints are very helpful. Let's say between agaricus, which are your portobellos, your button mushrooms, and your amanitas, right? Like,
Right.
That's a real clear difference between are we having lunch or are we going to the ER? But otherwise, I mean, it's also just like beautiful art. And there's another kind of practicality there. And I would mention, so this was my second round of doing them because I did them initially. And then, so here we are, it's a Tuesday. I had done it over on the Sunday night into Monday. And it's like, great, we got the spores. And then when I came back that evening, it basically almost all but disappeared. And I was like, well, that's not going to show up on the camera.
Because you took the lid off.
Yeah. So then I did it again and then kept the lid on. So that's a key point.
Or for teaching purposes, like I said, that's when I used to take
You
that and
lacquer
lacquer it.
Yeah.
And as long as it's depending on the type, you'd have to experiment. A hairspray
Right.
will work depending on the hairspray.
Yeah.
And then I'd have something in my binder.
Right.
Like it's still in that binder somewhere.
Yeah. That being said, I remember Paul Stamets telling a story. I believe he was in Australia and he found, I don't know if it was a psilocybin mushroom, like something that he was like quite interested in. And so he grows out mushrooms in his lab and he wanted to take specimens home, but you know, being international travel on a plane through customs, this and that, he just didn't feel like he wanted to officially declare
Right.
it and go through the whole rigmarole. So he did a spore print, you know, looked like a piece of art,
Yeah.
just tucked it into a book and brought it back home with him and then was able to kind of grow it out from there. Because as you
Oh, I see.
mentioned,
Yeah,
I
'cause
mean,
they're-
that's
yeah, they're
essentially
bound.
the seed, right?
Yeah.
Like this would. So these are just shiitake mushrooms from the store. What I do recommend is actually taking off the stem or the stipe, as Blaine had described it, because then you can get all that surface area onto the paper, at least for
right,
a
yeah.
nice kind of uniform spore print.
You wouldn't get a spore print without taking the lid off. Yeah, the stipe's got to go.
Yeah, and then it's just nice and flat. And then if it does have a veil or it hasn't fully opened up, make sure that it has, obviously. Otherwise, it will not release onto the paper. And like you said, anywhere from like, you know, in as quick as within an hour to several hours to overnight, cover it, and that does it. And I like your trick because, you know, as you mentioned, there can be white spores, they can be brown spores, having two different colors of paper. Or you had mentioned in the past that sometimes you'd use magazine paper.
Well, I got confused that you had me along one night with the local society, and I overheard people talking about needing special paper.
Right.
And I thought, wait a minute, I've done hundreds of spore prints on the back cover of magazines.
Yeah,
just
yeah.
where there's a cigarette ad or something with
Right.
big black and white letters.
Yeah, yeah.
And then you
'Cause
can
you
just
got both the colors right there.
yeah.
Yeah, because the idea is you don't know if it's going to be white. You don't know if it'll be brown, like a dark color. So having that contrast, or you had said you had done half and half, underneath the cap.
In a
Makes
container.
sense. Yeah. Cool. So simple process. I think it's a fun little piece of art, to do it with the kids, that kind of thing. And then it may come in handy as you are trying to key out different species, as you're looking through the book. Does it have dark spores? Does it have light spores? And as Blaine said, that can also be a starting point. Okay, white spores are these, dark spores are there, and then you can keep going down.
And so back to using the keys and just some other back to descriptive adjectives that you need to learn for green plants, or if like it never stops with leaf shapes and so forth. So when you slice open a gilled mushroom, you look at the gills from the side, and they may be attached at both ends, the tip of the fruiting body to the stipe. So they can be attached, or they can be free swinging at the stipe end, or they can have a little notch in them. It's called adnexed. And so those are further clues that when you get to the key, once you're into, so a genus, species is always the last thing. Do you need to go?
Yeah, Laura's just going to grab that. So
Okay.
That was,
So you get to genus quick enough, and then sometimes you need those last clues to get to species.
Right.
Like another genus with gilled mushrooms is if you take a little pocket knife and slice the gills, they may ooze a latex.
Right.
It's just like the latex from a dandelion stem. And so that genus is called Lactarius.
Yeah.
And then you go to secondary characteristics. So there's one that
This book was really great for that, actually. I was looking at all the different type of Lactaria species they had.
they might stain or not.
Yeah.
And there's
right?
one that's
What's the color? Yeah, here it is.
a gal that I lived with for many years.
Does it have an odor? Is it strong? Is it lacking?
Yes, and then we get into smells.
Yeah, and they've even got one here, taste. Cinnamon, taste mild, becoming acrid.
Right.
Using all your senses.
So the bottom line then goes to when you're trying one for the first time, don't eat a lot.
Right.
And that was one that I made a mistake with. We were on one canoe trip. It was the only time that Miller let me down. It was with one of the coral mushrooms, and they were abundant.
Yeah.
And it said if it stains, I forget whether it was blue or brown, because I'd eaten them before. And they taste like it's like lobster.
Yeah.
If you cook them up with garlic butter, it's just so yummy. If you were blindfolded, you'd think of the texture and the taste. You think you got lobster there.
Yeah.
And I just loved them long before I knew they were medicinal.
Right.
But it was one of the coral mushrooms, and he said stained, but he said brown, and they stained purple, or the other way around.
Right.
And I thought, no, we're okay.
Yeah.
And so it's almost all we ate for supper that night because the mushrooms were so good.
Yeah.
We all ate a bunch of walleye fillets. We just had fish and mushrooms and full bellies. And the next morning, it was just my wife and my brother and his wife. We all had really bad diarrhea.
Oh, gosh.
And one of the things we had to do at the end of the morning was run a fairly big set of rapids to continue on our trip.
Oh, no.
So what do you do with, I was hearing in my imagination, Howard Cosell, I think his name is the moderator for a lot of sports.
Yeah.
And as if he was in a helicopter following us.
Yeah.
I could say, so what's going on here now? The lead boat's into that that was a big wave. And we can see the ballast person. Oh, it's a woman. What's she doing there? She's dropping her rain pants there, and that's a messy rock.
Yeah.
So running rapids with diarrhea is not a good day.
No, for sure.
And so whenever you try any new wild mushrooms, just put the rest in the fridge. If you have one, keep them on ice, and just have a few.
Yeah.
And see how they fit with you.
Yeah, exactly.
Before you pig out on them.
Yeah. Yeah, totally.
There's one that's in the Caprinus That's shaggy manes.
Right. Yeah.
And there's a common one that grows in lawns and such. Fairy rings. People, there's another
Right.
thing.
Yeah.
People ask me about what does fairy rings mean? I go, absolutely nothing. It's a new mycelium and it's growing out.
Yeah.
And you'll notice if it's in a lawn that the lawn inside the ring is healthier than the lawn outside the ring because it's actually leaving behind some extra nutrition
Right.
for the lawn. So, and then the fairy ring might get bigger and bigger and bigger. And if you were, say, on a farm, you'd have to be looking from the air, looking with one of those little helicopters. You might see that the ring was getting bigger and bigger and then hit the chicken coop. But this part kept growing. And then it came over here, and then one part of it hit this big rock.
Yeah.
So eventually you don't realize that the fairy ring is now half a city block wide,
Right.
but all these objects got in the way and it kept growing anyway.
Yeah. Yeah.
So I don't think fairy rings mean much
Right.
in terms of safety or any of that.
No. No.
But
Yeah.
uh,
Yeah, generally associated with a particular species. And then again, it has its own identifying characteristic. Okay, it's got gills. And a lot of these books, it's kind of like asking those questions, and observing.
so Caprinus that's where the shaggy manes come in.
Yeah.
And if you're ever picking that mushroom, they're kind of a tall cone shaped, sort of black and white with all like Farrah Fawcett hair. And they haven't, it's a word that I've never seen mean anything else in the English language,
Oh, yeah.
where once they're picked, if they get abused at all, they release enzymes
Yeah.
that they break themselves down.
Yeah.
It's called, here's the word, autodeliquescence.
Yeah.
What does that mean? Digest yourself.
Yeah.
So if you throw them in a bag and throw them in your pack and you're hiking, by the time you get around supper time, you have a bag of black ink.
Yeah.
So some of those you have to pack very carefully in a basket,
Yeah.
which I saw everybody that night at the club was picking in baskets, right?
Yeah.
Not in in fanny packs and stuff like that.
Yeah.
So until you get to the identification phase, really you shouldn't even put. I didn't want to outstep my place there, which I had no place there, other than to watch. When you're picking a whole bunch of species in a basket, even that is potentially troublesome. You should have a bit of wax paper or something to separate different. If there's species you don't know,
Right.
keep them separated because
Yeah.
they're moist.
Yeah.
And some of the toxins, if there were any, could transfer.
Yeah. Yeah.
Okay,
No, it's
right?
true.
Keep them separate.
Yeah. Good. Well, I think that gives everyone a good sense of, as we started off, like keying out. It's a great kind of conversation and reference of like plants keying out, which, is equally applied to mushrooms, can be a process, a little bit of a learning curve to start. But as you shared, you can kind of like fast track your way once you start to know the terminology and then certain characteristics of certain species. And then it's less flipping through every page trying to, pinpoint it from there.
And another thing, this has happened to me because I've eaten a lot of the Aspenbolides. I used to love finding them up on the Churchill where I was camping. The size of pumpernickel bread.
Right.
Like they get
huge.
this big, right? And in 2 days, like
Yeah.
they grew that big almost overnight. And somehow, flies can get into them real quick.
Yeah.
So if I'm gonna bother to fill a bag full of stuff and go home and key it out,
Right.
I always break one open first and make sure it's not crawling
Yeah.
with
for
worms
sure.
already.
Yeah, those ones in particular are notorious through the stem, that kind of thing.
Unless, if you're gonna cook them really well, I suppose there'd be protein content there. But
Yeah.
I've never stepped that low.
Yeah, for
and
sure.
some other ones.
Yeah.
So
Okay. Well, there we have it. Yeah. Thanks for bringing your books and resources and teaching us all about it.
this much about it.
All right.
There's a lot to know,
Yeah, that's right. It's a lifetime.
but we try.
Yeah. Okay, well, bye for now.